Hello everyone, it's Mimi here. BMS working in the NHS. And today's video will be about tissue selection, which is part of the IBMS specialist portfolio in cellular pathology. So, just a quick introduction to this video. It will be short and sweet, I promise. I will discuss rationale and aims, learning outcomes, indicative content, resources, submission of evidence, Q&A ideas, evidence ideas, reflective statement, and some helpful tips to make candidates' portfolio better. With all that said, I hope you will enjoy the content of my video and I hope you will learn a lot. So let's dive into this submodule. Rationale and aims of this submodule outline the general information and expected knowledge and competence that candidate needs to be achieved when this submodule is completed. These are relevant to each submodule. For this submodule, the expectations are that the candidate will be able to recognize, apply and appraise the importance of maintaining the link between the sample and its request form. Work in a safe manner during tissue transfer of Category A. Discuss and employ measures to avoid tissue loss, sample mix-up and contamination. Understand the impact of poor orientation during tissue selection and its consequences to subsequent procedures. Understand the impact of pre-analytical errors relevant to tissue selection submodule. Provide examples of troubleshooting relevant to tissue selection submodule. The learning outcomes demonstrate candidates' knowledge, skills, and competence in this submodule. They also include demonstration of the understanding of scientific principles, laboratory procedures, quality assurance, and professional conduct. They allow candidate to work autonomously and solve problems independently. The first learning outcome is discuss the importance of the use of identifiers on the request form and containers to match, in avoiding sample mix-up. Candidate needs to be able to discuss the importance of using identifiers to avoid sample mix-up. Does the candidate know why it is important to check patient identifiers on the sample pot and the request form? Do they check the relevant information? Do they know how many identifiers are needed to accept the sample for analysis? Do they know what to do when the patient identifiers do not match? Do they know the consequences of sample mix-up? Candidate could consider patient safety and fatal errors, wrong diagnosis, treatment, operational efficiency and sample rejection, cost management, quality assurance, delays, repeat testing, increased workload, clinical risk mitigation, legal and regulatory compliance, percentage of errors occurring in pre-analytical phase, minimum data set, key identifiers, use of technology, best practice, consequences of poor sample labeling. The second learning outcome is, demonstrate how your laboratory operates to maintain the link between sample, cassette, and request form including labeling and storage methods. Include how specimens are logged on laboratory information management systems and/or tracking systems appropriate to your own laboratory. Candidate needs to demonstrate procedures for maintaining the link between sample, cassette, and request form. Does the candidate maintain the link between sample, cassette, and form throughout the lab correctly? Do they correctly use labeling system? Do they correctly enter samples onto the LIMS system and track them throughout the lab? Candidate could consider LIMS system used, tracking system and accessibility of data, barcoding, blocks, cassette and slide labeling, storage for samples pre- and post-dissection, date and time stamping, data entry, safety measures throughout this process, percentage of errors occurring in pre-analytical phase, link between the pot, form and cassette, information verification during dissection, SOPs, audit trail, traceability, possible errors, and how they can be minimized. The third learning outcome is, recognize the hazards associated with tissue selection and explain the appropriate actions to maintain safe working, including steps to take in the event of an incident. Candidate needs to recognize the hazards of tissue selection and maintain safe working. Does the candidate know what possible hazards are associated with tissue selection? Do they maintain safe working environment during tissue selection? Do they take relevant steps to correct incidents? Candidate could consider general biological, chemical, and physical hazards, cross-contamination, aerosol generation. Safety measures like vaccination, precautions, PPE, engineering controls like ventilation, safe handling risk assessment, cleanliness. Possible incidents like sharps or splash injury, biological spillage, fume inhalation, sample mix-up, steps they would take to deal with those incidents. The fourth learning outcome is, demonstrate how you would set up the dissecting workstation in an ergonomic manner ready for dissection. Candidate needs to set up a dissection bench. Does the candidate know how to set up dissection bench? Do they know why it is important to set up dissection bench in an ergonomic manner? Do they know consequences of not setting up the bench in ergonomic way? Candidate could consider equipment needed, position of equipment, setup of equipment, ability to set up the chair and bench ergonomically, feet positioning, knee gap, shoulder position, arm support, working zones, lighting, accessibility to sink or bin, place for specimens, microbreak, consequences of non-ergonomic setup, and so on. The fifth learning outcome is, demonstrate with examples from your practice the transfer of Category A specimens from pot to cassette relative to your own scope of practice, maintaining appropriate audit trail records. Candidate needs to transfer Category A specimens. Does the candidate know how to perform transfer of Category A specimens? Do they maintain the audit trail during the transfer? Do they record relevant actions during the transfer? Do they know where to record information relevant to specimen transfer? Candidate could consider scope of their practice, HNS requirements, quality assurance, equipment, LIMS and tracking systems, SOPs and methodology used, checks performed like checking patient identifiers, sample description, dictation, sample handling, orientation. Specimens with special instructions. Documentation, scribing and recording information for traceability. Possible sources of error and how they can be prevented. The sixth learning outcome is, discuss the implications of specimen contamination and missing tissue and demonstrate with examples measures taken in accordance with laboratory protocols at specimen transfer to avoid specimen contamination and tissue loss for subsequent procedures such as processing and microtomy. Candidate needs to discuss implications of specimen contamination and missing tissue. Does the candidate know the implications of specimen contamination or tissue loss during tissue selection? Do they take appropriate measures to minimize contamination and tissue loss? Do they know how to deal with specimen contamination or tissue loss? Candidate could consider implications of contamination and tissue loss on subsequent procedures and reporting like diagnostic errors, therapeutic errors, patient safety, operational impact, ethical consequences, legal risks, reputation. They could consider measures like cleanliness, careful approach, following SOPs, disposable tools, careful sample handling, one sample at a time, sift through instead of transfer, use of sponges or spicy wraps. The seventh learning outcome is, explain and apply the importance of correct orientation of samples at specimen dissection and state methods taken to distinguish them in subsequent tissue preparation procedures such as embedding and microtomy. Candidate needs to explain and apply correct orientation of tissue. Does the candidate know what the importance of correct sample orientation during tissue selection is? Do they know the impact orientation has on subsequent procedures? Do they know the implication of incorrectly orientated specimen? Do they know what sample types need to be orientated during tissue selection? Do they communicate the need for orientation correctly? It could consider sample types needing orientation like skin or tubular structures, reasons behind tissue orientation, ways of tissue orientation, ways of communicating orientation instructions to the embedders and cutters, use of ink, printing information on cassettes, instructions within tracking system and request form. The eighth learning outcome is, demonstrate with examples troubleshooting problems which may arise pre- and post-analytically, including root cause analysis. Candidate needs to be able to troubleshoot. Does the candidate know how to troubleshoot possible pre- and post-analytical problems that can affect tissue selection? Do they take corrects? Do they know the implications of pre- and post-analytical errors? Candidate could consider pre-analytical errors like mix-up of samples, specimen misidentification, incorrect sample labeling, sample sent dry, sample lost in transport. They could consider post-analytical errors like delayed reporting, transcription error, result interpretation or misdiagnosis. They could consider troubleshooting steps like error identification, source of error identification, corrective and preventive actions, communication, and so on. Those are the learning outcomes candidate needs to meet for this submodule. Indicative content highlights background knowledge that might be assessed during the Viva. They may or may not be demonstrated by the learning outcomes or candidate's evidence. In this submodule, candidate is expected to have the knowledge and understanding relevant to situations when specimen contamination may arise and how to avoid them. Equipment, technique, environment. When tissue may require further or alternative fixation methods. Candidate could consider samples for molecular, EM or F analysis. Hemorrhagic tissue, high-risk specimens, tumor resections, etc. Candidate could consider procedures used for those samples. Indicative content also highlights skills and competence the candidate is expected to be able to do. For this submodule, candidate must be able to set up dissection bench in preparation for tissue transfer and those performing specimen dissection. Assist in specimen dissections with a range of dissectors and take or set up equipment used for dictation. Follow SOPs, error logging. Give accurate macroscopic description of Category A specimens supported with audit relative to own scope dis. Take appropriate action in events such as the reporting of missing tissue at tissue transfer in accordance with lab protocol. Identify tissue types that require specific orientation at dissection and communicate clearly to the embedder and microtomist the importance of the correct orientation of epidermis, lumen, serosa, and mucosal walls. Operate and perform basic maintenance on cassette labeling equipment. Resources section on the Brightspace contains some very useful information for this submodule. Submission of evidence section on Brightspace informs the candidate what evidence needs to be submitted for each subsection. Candidate needs to submit answers to questions that were set by the training officer. I will share some examples of possible question suggestions later on. Candidate also needs to select an additional piece of evidence relevant to this submodule. For evidence ideas, please have a look at the video for trainee. Each submodule also requires the candidate to submit their reflective statement relevant to their practice and training in each submodule. These are some example questions you could give your candidate to test their knowledge, competence, and skills about this submodule. These should be based around the learning outcomes mentioned earlier. These are some more example questions you could use for this submodule. You could ask them to complete tables you created based on the learning outcomes. Or you could use images to test candidates' knowledge and competence. Supporting evidence should be selected by the candidate. It needs to be based around the learning outcomes and indicative content, and be relevant for each submodule. The evidence should allow the candidate to demonstrate their specialist knowledge and competence in each submodule. It should be specific and relevant to the candidate's in-house practice. If you would like to see specific ideas for supportive evidence, please have a look at the video for trainees. Each submodule also contains reflective statement, which is asking the candidate to summarize their role in the lab in the context with each submodule. How do they demonstrate that they can draw a conclusion from what they have learned, reflect on their current practice and how it can change in the future due to gained knowledge? They could think about what is their lab role in context of this submodule. What impacts can their actions have on their colleagues, department as a whole and for the patients? They also need to write a personal reflection on their training in the lab and explain why they chose their evidence. This is very personal to their own circumstance. It should demonstrate their ability to be self-aware when analyzing gaps in their knowledge. They can use it as an opportunity to reflect on aspects of their training, application of their gained knowledge and skills, and how they have achieved their goals. They should think about what their evidence demonstrates and why they chose this specific evidence for this submodule. Also, they should write about what their training was like when it comes to this submodule and how their practice changed due to their training. Their reflective statements can also be used as a CPD evidence in case they are chosen to be audited by the HCPC. I would also like to share with you some helpful tips that can make candidates' portfolio look really good. Ask them to try and present the information in various formats such as tables, animations, pictures, posters, flowcharts, videos, photographs, PowerPoints, diagrams, graphs, infographics, charts. This nicely breaks down a lot of text that is usually present in specialist portfolios. Ask them to try and make it a little bit more interesting to the reader, which will be, and eventually the assessor. Ask them to try and put themselves in your or the assessor's shoes. How would they like to read hundreds of pages of plain text? There is a saying I really like, and it goes like this: a picture is worth 1,000 words. I will make a video where I will share with you my evidence I used for my portfolio back in the day. Hopefully that will help. Also ask them to try and use various types of evidence, not just one type, like a case study, for each submodule. They could use experiments, case studies, annotated results, troubleshooting, controls production, annotated images, risk assessments, glossary, training presentation, certificates with reflective statements, nonconformances, validations, verifications, comparisons, ECHA results, annotated audits, kappa or DATIX, Q&A. Ask them to try and bring in their personality. Ask them to think about the ways they like to learn best. If they like experimenting, let them perform experiments. If they like troubleshooting, let them perform troubleshooting. Ask them to choose that demonstrates their specialist understanding, knowledge, and skills, and how they apply this in their practice. And one more thing, ask them to keep the learning outcomes and indicative content in mind when they are selecting their evidence. Learning outcomes that require the candidate to demonstrate something would be ideal for covering with their evidence. And this brings me to the end of today's video. Hopefully you now have a better understanding about the learning requirements for this submodule. And I hope you also have a better idea what questions you can choose to test candidates' knowledge. And my final take-home message? Keep those learning outcomes in mind when selecting questions and guiding the candidate.